Welcome to The Explainer. We hear a lot about AI these days, right? It's improving our search results, it's powering cars, but today we're talking about something way bigger. We're exploring how AI is being used to unlock the absolute fundamental secrets of life itself, and how one single breakthrough is pretty much changing the course of science forever.
So get this. Every single thing your body does, everything from digesting your lunch to fighting off a cold to you just moving a muscle right now, it's all powered by trillions of literal nanomachines. These microscopic little workhorses of biology, they're called protein.
So, how does a protein actually work? Well, your DNA, your genetic code, gives the instructions for a long chain of molecules, kind of like beads on a string. But here's the magic trick. That simple string, spontaneously, all on its own, folds into a very precise 3D shape. The lead scientist on this project, John Jumper, he has the perfect analogy for it. It's like an IKEA bookshelf that builds itself. And it's that final, folded shape that allows the protein to do its job.
Which brings us to Section 1, Biology's Grand Challenge. Because figuring out that folding process, that's been a problem for a long, long time.
For half a century, predicting how a protein is going to fold has been one of biology's biggest, most stubborn problems. And here's why it's so hard. This folding, it doesn't happen in some clean, sterile lab. A cell is an incredibly crowded place. Jumper, the scientist we just mentioned, he compares it to a public swimming pool on the 4th of July. Just total chaos. And somehow, in the middle of all that, this delicate little string has to find its one perfect shape. And that right there is the grand challenge. How does this simple, one-dimensional line of code from our DNA reliably produce a super complex three-dimensional machine? If you could crack that, you could revolutionize how we understand diseases and design new medicines. It's a huge deal.
For decades, the only way to see a protein's shape was through these painstaking experiments. It was so hard, in fact, that for just one protein, it once took a team of scientists over a year of failed attempts just to get the crystals they needed to form. A full year for one structure. There had to be a better way.
So that brings us to Section 2, a scientist's winding journey. Because the story of the person who solved this is almost as interesting as the problem itself. The guy who led the charge was John Jumper, and his path was not exactly straight. He started out as a physicist, but then he dropped out of his PhD program. He got a job in computational biology that sparked a whole new passion, which led him to a different PhD, and then eventually to Google's AI lab, DeepMind. It was this really unique mix of skills that gave him the perfect perspective to attack the folding problem from a new angle.
Okay, so let's get into the good stuff. Section 3, the breakthrough recipe. How did they actually do it? So what was the secret sauce? It really came down to three things. First, data. They had about 200,000 protein structures from a public database. Second, compute. Powerful computers, sure, but not some unimaginable, enormous system. But the real game changer, the secret ingredient, was the third thing, research. Just better ideas.
And you might ask, just how valuable were those new ideas? Well, another lab ran this amazing experiment. They took the new version, AlphaFold 2, and they trained it on just 1% of the data. And it was still more accurate than the original version that was trained on all of the data, 100% of it. The takeaway here is stunning. The right research ideas were more powerful than a 100-fold increase in data.
So what was the big magical idea? Well, the truth is there wasn't just one. A lot of people pointed to this really clever concept called equivariance. It's basically teaching the AI that a protein is still the same protein even if you rotate it in space. But, when the team actually tested this, taking that one idea out only hurt the performance a little bit. It wasn't the magic bullet everyone thought it was.
As John Jumper himself explains, the breakthrough wasn't some single giant leap. It was the result of combining many mid-scale ideas. Each one added another piece to the puzzle, and eventually they all added up to create something truly transformative.
All right, Section 4, putting the tool to work. Because look, building this thing was one thing, but what happened next,
That's what changed everything. The team didn't just publish a paper and call it a day. No, they used AlphaFold to predict the structure of nearly every known protein on the planet. We're talking over 200 million of them. And then they released it all for free in a public database for any scientist anywhere to use.
This is the moment the scientific community really went from skeptical to sold. Biologists started looking up the specific proteins they were secretly working on, things nobody else had seen. And they were just stunned. One of them actually asked, "How did DeepMind get access to my unpublished structure?" The AI had correctly predicted their results before they could even publish them. That's when they knew this was the real deal.
And the impact was immediate. One researcher who'd been struggling with a single problem for months solved it in a single morning using AlphaFold. Their reaction just says it all. "I want my time back." The AI was giving scientists back their most precious resource. And then came this shocking discovery from the community. Just 2 days after the code was released, a user tried something totally unexpected. They fed 2 proteins into the system at the same time, just to see what would happen. And incredibly, AlphaFold predicted how they would interact with each other, a skill it was never, ever trained for. This showed the AI hadn't just memorized a task, it had somehow learned the fundamental rules of biology. So that brings us to the final part, Section 5, the future of discovery. Because the true power of a tool like this isn't about replacing scientists, it's about amplifying their ingenuity.
Let me give you a perfect example of what I mean. Scientists were studying this amazing natural machine nicknamed the molecular syringe. And it does exactly what it sounds like. It literally injects other proteins into cells.
So they had this brilliant idea. What if they could re-engineer this syringe to deliver specific medicines directly to target cells, like cancer cells? The problem was they didn't know its structure, and they had tried over 100 different experimental approaches to re-engineer it, and every single one had failed. Then they ran an AlphaFold prediction. The result, even though it wasn't perfect, it revealed something absolutely crucial. The syringe had legs. This was the aha moment. The scientists immediately realized the legs must be how it attaches to cells. And right away, they knew exactly what to do. Just replace the legs with custom ones to target new cells.
And it worked. Thanks to that one AI prediction, they successfully built a whole new system for targeted drug delivery. They even tested it in a mouse brain. The AI didn't do the experiment, but it gave them the key insight that broke the logjam. It amplified their own creativity and let them solve the problem.
So where does all of this lead? AlphaFold absolutely transformed biology. But it leaves us with what might be the most exciting question of all. One John Jumper himself asks, how general will AI for science be? Can we see these kinds of breakthroughs in chemistry, in materials science, and climate science? We are just at the very beginning of finding out. Thanks for joining The Explainer.